The energy in a proton motive force is used to drive the synthesis of ATP. We just used oxidative phosphorylation to make ATP. There's some little summary things we need to sort out. When we talk about the coupling of a proton gradient with the making of ATP, we call that chemiosmosis. So a term that you're going to see is chemiosmosis, an energy coupling mechanism that couples the energy in a proton motive force, I'm just going to write PMF, with the making of ATP, the production of ATP. Okay, chemiosmosis is kind of this proton motive force and ATP synthase, making ATP, right? Whew. This whole process of making ATP in this way is called oxidative phosphorylation. So the drawing that I drew that includes the electron transport chain and the proton motive force here and chemiosmosis, this whole component here. This whole mechanism of making ATP is called oxidative phosphorylation. The making of ATP, or I forget how I repeat it. Let me just get out my notes here. ATP production driven by the transfer of electrons from food to oxygen. ATP production driven by the transfer of electrons from food to oxygen. Where's the food in the picture? NADH and FADH2 are made as a result of the oxidation of glucose. That's our food, really, right? So we've got these NADH and FADH2 donating electrons to the electron transport chain. Energy that's released is used to create a proton motive force that's used to drive the synthesis of ATP as protons diffuse across the membrane. So oxidative phosphorylation is simply the ATP production driven by the transfer of electrons from food to oxygen. If oxygen weren't here as the final acceptor of electrons, we couldn't run the electron transport chain. And we couldn't create a proton motive force. So we couldn't create ATP because we wouldn't have this proton motive force to do it. So in your notes somewhere, right, this whole process here is oxidative phosphorylation. We're going to have to wrap this up with follow-up videos. We're going to look at a ledger of what we've just made in the process of following one molecule of glucose through aerobic cellular respiration. We're going to do that in a different video. All right, thanks so much.